Good morning, everyone. Happy Friday. Don't we just love it? This day before the weekend, and you can sleep in tomorrow. It's just the best. So today, I sent an email around yesterday saying that today, for half of the time of the lecture, I will focus on the patterns of inheritance. And for the second half of the lecture, I ask you to go over the exam study guide and write down your questions that maybe regarding the material that you do not understand fully, or you don't know what I mean, what Ansley or I meant by including a certain point in the exam study guide, etc. Or related info regarding the course that is not completely clear to you. So today, for the second half of the lecture, we will focus on solely your questions. Okay, I posted the participation assignment for today, and for today, it's a Friday kind of participation assignment. It is a discussion. I'm asking you what kind of biology related news stuck with you, and what did you remember about it. I ask you to describe it in the gist in a couple of sentences and drop the web link. So if you remember a particular new biology related news, even if it's from 5 years ago, submit that. Okay. If you, I'm asking what stuck with you, but if you nothing really stuck with you, then you can go and search for biology related news. But I would encourage you to prioritize the news that did stick with you. Like I'm interested in your thoughts and what you think about, what kind of biology related information is interesting to you. So that's why I'm asking you to prioritize the news that stuck with you rather than just go and search the recent kind of news. All right. Any questions before I get started? Okay. All right, so a couple of reminders as usual. Quiz 3 is due today. Quiz 4 opens tomorrow, and you have all the time until Monday, February 22nd to submit it. And again, you can take quiz 4 anytime between tomorrow and Monday, February 22nd. The discussion submission is due today. And we have the additional assignment for which I extended the due date. This participation assignment is designed to test the lockdown browser, and it is due today. If Please figure out your lockdown browser by the end of the day today. If you haven't contacted UCF Web Courses support, I strongly encourage you to do so. Um, I am not a tech rep. Sometimes I don't, most of the time, I am not able to help you. Sometimes I am able to help you. For instance, somebody contacted me and said, oh, the Chromebook is not allowed to be used in this particular setting. So I went back to settings and I changed it. But if you're getting, if you're prompted to reinstall the lockdown browser, I cannot help you with that. The UCF web courses support person will be able to help you with that. If you're still having trouble after having contacted the UCF web courses support person, email me and let me know. And let me know what reason that UCF web courses support person gave you for your particular situation, why you cannot, why lockdown browser does not work for you. All right, so the topic of today's short lecture will be patterns of inheritance. And as a reminder, I think it would be useful to review what DNA is. And DNA, the function of the DNA is to carry our genes, is to carry all those recipes for lots and lots of proteins that we need to support our processes that uh, ensure our well-being. Recall, please, that a gene is a portion of DNA whose sequence of nucleotides, A, C, G, and T, or adenine, cytosine, guanine, and thymine, encodes a protein. And the proteins are expressed in cells, determine many characteristics of an organism, such as eye color, hair color, skin color. I'm focusing on colors here, but there are plenty of more traits or characteristics that the genes encode. Remember that in we and other organisms that reproduce sexually have two members of chromosomes in a pair, and these two members in a same pair, these two chromosomes that belong to the same pair are called homologous chromosomes. So members of a homologous pair have the same genes, but might have different versions or alleles of the genes. So these alleles may be identical. For instance, gene A inherited from the mother and gene A inherited from the father. You see that it's both capital A's indicating that these alleles are identical. These alleles will encode the same eye color or the same hair color or some other trait that is going to be identical. Or these alleles can be different. For instance, gene B here, or allele on mother's side, the allele is denoted as capital B, and on the father's side, the allele is denoted as lowercase b, which indicates the different version of a gene. For instance, if it's a capital B, let's say B stands for brown eye color. So the mother has brown color, but the father does not have brown color. For instance, the father has blue color, but lowercase b can denote that non-brown, okay? So often, so how are these new alleles created, you might wonder? And the answer to that question is going to be fairly simple, and what we've encountered before. The cause for new alleles is often it results from mutations, or new alleles result from mutations. And often mutations are changes in DNA nucleotide sequence. Remember, there can be substitution mutation, like that's the kind of mutation that causes the sickle cell disease. But again, most mutations are harmless, and they kind of expand the genetic diversity. Often these mutations cause altered proteins to be produced, and again, most of the altered proteins are harmless. So, the first person who uncovered basic laws of inheritance was an Austrian monk who was called Gregor Mendel. So he discovered the patterns of inheritance, or the basic laws of inheritance, before the DNA was even discovered as a molecule. So if you don't remember it or didn't know it at all, DNA was discovered in the 1950s. DNA as a molecule was discovered in the 1950s, but we have started like crop selection and like selecting dog breeds and whatnot much earlier than 1950s. So we basically employed the information contained in the DNA and mutations in the DNA before we even knew DNA molecule existed as such. So Gregor Mendel was a pioneer in the work of selection, and he liked plants and statistics. Those were his two passions. And through cultivating many generations of plants and counting the incidence of particular traits in those plants, he made important discoveries in genetics even before the genetics as a field was founded. So his plants of choice was peas. And the reason he liked peas is they're easy to control. It's easy to control their mating. If you want the plant to self-pollinate, you just cover the flowers up with a piece of cloth to ensure the lack of cross-pollination. And if you want the plants to cross-pollinate, you can basically do it manually. Even you don't have to rely on bees or wind. So another reason why Gregor Mendel liked pea plants is because they produced many offspring. So I assume you've all seen the pea plant. It's pretty common. And if you haven't seen the pea plant, you've definitely seen the pea pods. And, and one pea plant can have a lot of pea pods. So one individual can produce many offspring, and they're relatively fast growing. I mean, I planted a pea plant on my patio about a couple of months ago. I already have pea pods. Like I gathered my first harvest the day before yesterday. And by harvest, I mean 2 pea pods. Anyways, So, Gregor Mendel was an Austrian monk who was passionate about plants and statistics, and he did some first experiments with patterns of inheritance. So, the gist of his experiments was he basically allowed the plants to cross pollinate, or restricted cross pollination, and after the plants bore fruit, he examined the properties of the seeds and planted the seeds again to examine the properties of the second generation plants. Okay, so that's the high level overview of what he did. So first thing that Mendel noticed is that when he prevented cross pollination, plants did not always produce similar offspring. Some plants, well, he noticed the inconsistent patterns in the offspring. For instance, once when he prevented cross pollination in a pea plant that was that grew from a green pea seed, he noted that um, the pea pods produced by this plant were always contain the green peas. However, when he planted yellow peas and grew them into a plant and then examines the fruits of the plant, he noticed that sometimes yellow peas produced the plants that bore produced yellow peas as well. But sometimes when he planted yellow peas, the resulting fruit was differing in color. Sometimes it was yellow, sometimes it was one green piece, and sometimes there were two green peas in a pond out of four. Okay, so and that did not make really much sense to him because in the early days, if you plant a green pea, you should get green peas. If you plant a yellow pea, you should get yellow peas. So if you're getting green peas, well, something's not right. That's unusual. And being a scientist at heart, even though a monk, which monks did science in those days, basically. He got curious about it and decided to keep planting these peas and try to notice if any patterns emerge, if he can make any scientific conclusion about what causes this discrepancy, or why this discrepancy rather came to be between the fruits of the fruits of the plants that came from similar looking seeds. So a bit of a terminology here before I move on. So the plants that were planted, like the green peas that produce green peas and the yellow peas that produce yellow peas, are called true breeding plants. Those are plants that produce offspring identical to themselves. And the plants that are called hybrids, they outwardly resemble true breeders, but produce mixed offspring. So outwardly, the yellow peas, they resemble the true breeders, like they're the same color, but they produce mixed offspring. So they would be called hybrids. And as I will mention a bit later on, and maybe you'll know this already from the grade school, it's because this guy is heterozygous in their genes. So when Mendel started cross-fertilizing pea plants, he noticed some intriguing results. When he cross-pollinated green peas and yellow peas, sometimes he would get plants with all yellow peas, and sometimes he would get plants with some yellow and some green seeds. Um, so, but in the next generation, green seeds reappeared. So sometimes pods contained yellow and green peas. The main takeaway, or one of the main takeaway of his cross-pollination studies, was that the trait or color seemed to one trait or one color seemed to obscure the other, and he called the trait that was masking a dominant trait, and the trait being masked a recessive trait. So the dominant trait in this example is the yellow color of the seed, because when you cross pollinate, when you cross the green seed, when you do sexual reproduction between a plant grown from green seed and a plant grown from yellow seed, you may get all yellow seeds, or you may get like some yellow and some green. But the pattern here is that yellow color overpowers the green power. So the yellow color is considered to be a dominant trait. In this slide really calls the allele dominant and not the trait, and it is because we don't really use the dominant trait kind of expression anymore. Mendel did, but since that was before the DNA discovery and the whole field of genetics, uh, people used like dominant and recessive relative to traits. But now the expression would changed a bit, so we use the terms dominant and recessive to the terms alleles or versions of a gene. And as I mentioned just now, some alleles are recessive, meaning they are masked by the dominant allele. The phenotype or the trait that is encoded by that allele is masked by the dominant one. So in this example, the recessive allele is the allele that encodes that produces the green color, or rather produces no color, because the green color is simply green color in a pea plant is basically the absence of yellow color. So what is happening here is the yellow color is produced by a protein, okay, and the protein that is expressed because there's some gene in a pea plant that encodes that protein. But when the pea is green, it indicates that the protein in this case is just not expressed. The protein producing yellow color is not expressed. So this trait is recessive, or this allele is recessive. So when you cross the two plants, one grown from green seed and yellow seed, you will get mixed results. You may get basically mixed results. And I will get to Punnett squares later to show you like how to calculate the probability of getting the green seeds, or basically various alleles from knowing if you know the traits of the parents. So dominant allele, dominant may imply more widely spread, like dominant trait or dominant allele may imply that this particular trait is more widely spread, but that is not always true. An allele causing, for instance, achondroplasia or a form of dwarfism is dominant. However, we don't see a lot of people with this condition walking around. So that means that kind of illustrates that the dominant allele is not always widespread. And also allele causing Huntington's disease. And if you watch the show House MD, one of the characters in the show has the Huntington's disease, and she's very worried about it, but they explain in detail like what Huntington's does to you. So the allele causing Huntington's disease is dominant, but it's not widespread. We don't see a lot of people with the Huntington's disease. So that's another example of an allele being dominant, but dominant is not equal to widespread, logically. But allele that causes blue eyes in people is a recessive allele. But nevertheless, we see a lot of people with blue eyes. And this is an example how recessive allele is widespread, despite not being dominant. So back to the peas. Why do some plants produce both yellow and green seeds? The answer has to do with each plant having two alleles for each gene because of their homologous pairs of chromosomes. So how is it possible? Why yellow does not overpower the green? So to understand this, let me touch on the chromosomes again, homologous chromosomes and meiosis. Suppose the color is determined by A gene here. So as a result of meiosis, these chromosomes, each chromatid will be separated into a different gamete. So we have the gamete that will contain capital A, another gamete that will contain capital A, another gamete all gametes will contain capital A, okay? So let's say capital A is the yellow color. And to be fertilized, like the one gamete needs, a female gamete needs a male gamete, and a male gamete needs a female gamete in order to produce a zygote. So if the female or / male gametes will have the same allele, capital A, for yellow color, the zygote will have both double A, double capital A genotype, and it will have a yellow color. However, let's say yellow color is encoded by B here. We have capital B on one chromosome, but lowercase b and lowercase b on the paternally inherited chromosome. So since the yellow color is dominant, the capital B, the yellow color will mask, will overpower the expression of the green color, or rather the yellow color, the protein encoding yellow color will be expressed. And during meiosis, we will have four resulting gametes. Two gametes will have capital B, or will have the gene encoding yellow color, but two other gametes will have lowercase b, meaning the absence of encoding, absence of the recipe for the yellow color. Okay, so if these two gametes, if this particular gametes containing one of this chromosome, it meets another gamete containing lowercase b, it will produce an individual that is going to be green in color. So although a parent had yellow color and contains alleles for green color, the offspring of this parent can contain the green color of peas. That was a little long-winded, but I'm sure you've encountered this information before, so I'm treating it more of as a review. So a genotype that I mentioned. So a genotype represents an individual's two alleles for one gene. Okay, so there are two similar words, a genome and a genotype. So genome is a collection of all of the genes in the DNA, but genotype is individuals' two alleles for one gene. So it's basically two versions of a gene that any individual has. We have three types of genotypes. The first type is homozygous dominant, and it's when this case here, when individual has two dominant alleles for a gene. So capital Y, Y stands for yellow color, represents the homozygous dominant individual. We have heterozygous dominant. Heterozygous individuals have one dominant and one recessive allele, and the dominant allele will mask the recessive allele and will be expressed. So if an individual pea plant is heterozygous for the yellow color, the peas will still appear yellow. And finally, homozygous recessive is when individuals have two recessive alleles. So double lowercase yellow, double lowercase y here represents the recessive allele for that represents the lack of protein causing yellow color, and the peas will appear green. And the genotype is responsible for the phenotype because, again, the genes provide the recipe for the proteins, and the proteins are helping produce the phenotype, produce the appearance. So phenotype basically is the physical appearance, like yellow color in the pea seed. So the phenotype is determined by how alleles interact with each other. Our phenotype is also determined by what kind of proteins basically are expressed by particular genes. So to determine the genotype of the parent, Gregor Mendel in particular, he looked at the offspring of the plants, of the pea seeds that he planted. So individual with the same phenotype can have different genotypes. So for instance, this yellow seed and this yellow seed, they have the same phenotype. They both appear to be yellow, but this yellow, when crossed with the green seeds, they produce different kind of offspring. So as a result, we can deduce that this guy is homozygous dominant and this guy is heterozygous because it will produce the some green and some yellow pea plants, pea seeds. So, and I just mentioned that, I'm just including this for you to refer to later. So this slide basically explains the how alleles encode the traits, allele Y encodes the active enzyme producing yellow pigment, and enzyme is a protein, and that's why the seeds appear to be green, appear to be yellow. But during the mutation, allele Y, lowercase y, encodes an inactive enzyme and does not produce yellow pigment. So I'm not going to repeat that again. It's just for your notes to keep for later. And finally, I'm getting to the Punnett squares. Punnett squares, which allow to deduce what kind of genotype the children will have if we know the genotype of the parents. So with our old friend peas here, a heterozygous parent plant for the yellow color will produce two types of gametes, a capital Y gamete and a lowercase y gamete, because these homologous chromosomes here will be split during meiosis. And another heterozygous plant will produce those are two female gametes, and another heterozygous plant, let's say, will produce two male gametes, which will be basically the same. There will be one gamete with a dominant allele and one gamete with a recessive allele. As a result, within the Punnett squares, we just cross all possible combinations of the male and female gametes. So we will get 4 possibilities, technically 3 possible outcomes, but it's helpful to write out the same outcome twice, the same zygote twice, in order to calculate the probability of getting a particular genotype and / phenotype of an offspring. So if both parents are heterozygous for yellow color, there is 25% chance of getting an offspring being homozygous dominant. There's 50% chance of getting an offspring that will be heterozygous, and there is 25% chance of offspring being homozygous recessive, right? But if we're looking at the phenotype, there is 75% chance, 3 out of 4, okay, 3 divided by 4 multiplied by 100, that's going to be our probability. There is 75% chance that the offspring will have yellow, offspring of 2 heterozygous plants will have yellow color. Okay, and that's the Punnett squares for us. Mendel's work can be applied to many traits, not just pea plants. So Punnett squares can be used and are used to track the inheritance of genetic disorders such as cystic fibrosis. And cystic fibrosis is a disease that thickens the mucus in your lungs, and it complicates your breathing. And also it affects your digestive system a bit. It's making it harder to digest things. So cystic fibrosis, the allele for cystic fibrosis is recessive, and couples can find out that if they're both carriers of the allele, and if they're both carriers for the allele, then they can estimate the chance of what probability is there of having a child who will have the disease. So here's our Punnett square for the cystic fibrosis disease. Let's say both parents are heterozygous for the fibrosis gene, and fibrosis allele is the allele that will cause fibrosis is denoted as a lowercase f. Okay, so if we write out the gametes, the genes in the genotype of the gametes, and produce the cross within the Punnett squares, we will see that there is 25% chance of producing a child who will be healthy, non-carrier, or homozygous dominant for the allele that does not cause cystic fibrosis. There's 50% chance having a healthy carrier, meaning a child who will not have cystic fibrosis, but will carry the gene for cystic fibrosis. And there's 25% chance, or one out of 4 chance, that of producing a child that will have the disease. Okay, and if you've had statistics before, you can do some basic math in your head. Like if parents have 2 children, what is the probability of 2 children having cystic fibrosis? If the probability of 1 child having cystic fibrosis is 25%, and the answer to that would be you would multiply 0.25 by 0.25, or 0.25 to the power of 2, and the resulting number will be like 6%. So having 2 children with cystic fibrosis is pretty low. And what is the probability of having 5 healthy children? So if parents decide to have a lot of children and are wondering, well, what is the probability that all of them are going to be healthy? Not one child will have the cystic fibrosis disease. And you would have to, so the probability of having one healthy child is 75%. But to get the probability of all 5 children being healthy, you would need to raise 0.75 to the power of 5 or multiply 0.75 by itself 5 times. And that would be around 25%, I think. Right? I'm going to check with my notes. 24%. So 24% chance that if you have 5 children, all of them will be healthy. None of them will have cystic fibrosis. So that kind of low in my opinion, but yeah. Some math to help you understand what you can do with the information if you and your partner decide to get genetic testing for some diseases. So there's some other things that I want to mention about patterns of inheritance, and one of the things is incomplete dominance. And this type of incomplete dominance creates a new phenotype. So the illustration I have here are red flowers and white flowers. And once like the red flowers are produced by, let's say, R1 alleles, and so those are homozygous dominant, those are homozygous dominant, but when they, homozygous recessive, or is it heterozygous? Anyway, the point is, when the 2 gametes cross with each other, they produce the heterozygous individuals, and these heterozygous individuals are not red and are not white, they're pink. So whenever whenever there's no complete dominance, whenever there's no trait that overpowers another trait, we are observing incomplete dominance. So one flower was red, another flower was white, but when 2 of them crossed, none of the flowers are either red or white. All of them are pink. So an example how heterozygous individual can carry both phenotypes, carry both traits. And another thing I wanted to mention is that sometimes one gene can influence many phenotypes. And whenever one gene influences many phenotypes, it is called pleiotropy. The example of pleiotropy is the single mutated gene that encodes a defective connective tissue protein. And this situation causes so-called Marfan syndrome. And people with Marfan syndrome often have long limbs. They're very thin. They often have issues because they have this faulty gene that encodes a faulty protein. They have issues with connective tissue. They have issues with the connective tissues. Pardon the rhyme. Inappropriate one at that. So they have problems with eyes. They often develop cataracts and lens dislocation. They have problems with bones and joints. They have excessive growth in joints, and they have like this trait of sunken chest. They have problems with heart and lungs, which like may have stretched aorta, problems with the valves because valves consist of the connective tissue, and they often experience collapsed lung. And finally, I want to talk about the sex-linked genes because I'm going a little bit over. So the sex-linked genes have unique inheritance patterns. Sometimes the males inherit a disease and women do not, and that's because men and women have different chromosomes in the 23rd pair of the chromosomes. Women have XX pair, and the men have XY pair of the chromosome. So because of this difference, the diseases that are carried in one of the X chromosomes, they can be expressed in males, but not expressed in females, because males only have one X chromosome in the 23rd pair, and females and women have 2 X chromosomes in the 23rd pair. An example of such disease is good Lord, what is it called? Hemophilia. There we go. Hemophilia is an example of such disease. So the hemophilia is carried on the 23rd, the gene for hemophilia is carried on the 23rd X chromosome, on an X chromosome in the 23rd pair. So if women basically do not get hemophilia because they always have two X chromosomes, but males can get hemophilia because they have only one X chromosome. So if they inherit the recessive allele on the X chromosome, there's no second X chromosome to kind of overpower the uh, this this recessive allele. Okay, and you can examine the Punnett square later. I don't want to go over. I have a couple of slides, but I'm not going to go over them. I'd rather dedicate this time to your questions. So with that, I'll hand it over to you guys. Fire away.
I have a quick 2 questions, real quick.
Yeah, go ahead.
So for all of this parent patterns of inheritance and stuff, is this going to be on this exam or the next exam?
No. That's going to be on the final exam. So it's not going to be on the next exam because the next exam is going to focus on next units. So this is going to be all the way in the final.
And then what about meiosis and mitosis? Because I know it wasn't on the study guide when I was studying yesterday. So I was wondering.
If it's not on the study guide, it's not going to be an exam.
Okay, sweet. Thank you so much.
Yep.
Can you go over dehydration synthesis and hydrolysis again?
So dehydration synthesis is it was covered in the supplementary videos, and dehydration synthesis is when you have two smaller molecules, and these smaller molecules, one molecule contains the hydroxy group, another molecule, or OH group, another molecule contains some H atom. And dehydration synthesis is when one H from one molecule and OH from another molecule get removed, forming a molecule of water, and the two smaller molecules get linked into a larger molecule. So dehydration synthesis is also a polymerization reaction. So we're building larger molecule from smaller building blocks and creating the molecule of water in the process. Okay, and hydrolysis is basically the opposite. It's when the larger molecule is broken down into smaller molecules in the presence of water and usually enzymes. So for instance, the example of hydrolysis would be the breakdown of starch or cellulose into glucose. Both starch, molecule of starch and molecule of cellulose are built from the glucose, but they have different spatial structure. So in the presence of enzymes, both starch and and cellulose can be broken down into glucose, molecules of glucose, in the presence of water. Did that answer your question, Madeline?
Yeah, that was very helpful. I have one more quick question. For our quizzes, are those questions likely to pop back up on the exam?
Sometimes they will, sometimes they won't. Oh, speaking of quizzes, you will benefit from taking them early, not only because you have the opportunity to schedule office hours with me and ask questions about things that are not completely clear, even after having used up one attempt. But another reason why to take quiz early, you will have the access to the correct answers for the quiz for longer, because the access to the correct answers stops after a certain time, and usually it's like a couple of minutes after the deadline, after a couple of minutes after the quiz is due. Okay, other questions?
Could you go over what you mean by sources of carbon in plants, please?
Sources of carbon in plants. Okay, can anybody address this question? Like, what is the source of carbon for plants?
So that one, I believe it's the one about, I'm blanking for a second. I just had it in my mind. That's the one, I think it's the carbon and light reactions, if I'm not mistaken. I'll go back and look at the the files and the PowerPoints, but I'm pretty sure it's referring to the carbon.
And Ansley, I know what you're talking about. The clue is fine. The
Okay.
clue is fine. Yeah.
Okay.
Let's think about sources of carbon in plants. Where do plants get their carbon from?
The air.
Excellent, Dylan. Exactly right. So plants get their carbon from the air. They suck up CO2, carbon dioxide, from the air. They get water from soil. Remember, they're pulling water from the roots, from the soil, to the leaves. And as a result, in the presence of light, they're making sugars. So the source of carbon is CO2 in the air. All right, other question? Lauren, did that answer your question?
Yes, thank you.
All right. Who else? Just unmute yourself and ask the question because the chat is getting a bit overwhelming. Ansley, could you
I'm trying
please?
to as I can.
Okay.
I have a question. It says on the study guide, it says transport between plant cells, and I wasn't sure like what that really means. I kind of forgot.
That refers to the cell structure lecture, and I mentioned that plant cells communicate with each other in some way, and the animal cells communicate with each other in some other way. So plant cells, they when you look at a plant cell under a microscope, the plant cells are not completely isolated from each other. They contain these little channels called plasmodesmata, like these little holes in the cell wall and membrane that allow plants to communicate with each other. And in animal cells, it has to do with the protein, a particular type of protein junctions. Okay. So that's what the study guide clue meant about communication between plant cells.
Professor, will you mind go over the Krebs cycle, please?
The Krebs cycle. Oh, okay. Hold on. Krebs cycle. Where are you? Okay, Krebs cycle. Derivative of pyruvate is oxidized, CO2 is released, more energy is transferred to electrons. So we have, during respiration, we have 3 stages of respiration, okay? Glycolysis that occurs in cytosol, Krebs cycle that occurs in the mitochondria, and electron transport chain. So during the Krebs cycle, I did not go into much detail about Krebs cycle, and you will not get asked a detailed question as well. But the point is, the Krebs cycle does not use oxygen, and what it does, it produces some molecules of ATP, not much. It produces CO2, and it donates electrons to electron-carrying molecules. And the electrons are also donated during the glycolysis, and these electrons are carried all the way to the electron transport chain, which also uses oxygen and produces the most ATP and some water. Okay, so what you need to know about Krebs cycle is that it does not use oxygen. It occurs in the mitochondria. It produces some ATP, but not much. And it donates electrons to the electron-carrying molecules that are then carried to the electron transport chain.
So the electron transport chain produces the most ATP.
That's exactly right. I have a slide about this. There we go. So aerobic respiration yields 36 ATP, and most of those ATP molecules come from electron transport chain. So glycolysis produces 2 ATP molecules, Krebs cycle produces 2 ATP molecules, but electron transport chain yields 34 ATP molecules. So electron transport chain is the powerhouse of the cell.
Can you go over what the study guide means by organic compounds, be able to name all of them? Isn't an organic compound just a compound with hydrogen and carbon?
Yeah, but there are 4 major organic molecules. that I mentioned during the lecture. So it's carbohydrates, proteins, the nucleic acids, and
lipids.
Lipids.
Okay, okay, thank you.
A good way to remember that one is to remember the types of molecules that make up the components of the cell. So like DNA, your membranes, proteins, all that good stuff.
That is a good point,
Okay.
Ansley. Yep.
Thank you, Ansley.
Yeah, of course.
I have a question. Do we have to know about the base pairings between RNA and DNA, or just for both of them separately?
All of them.
Okay. Thank you.
Do you mind going over polar versus nonpolar covalent bonds?
Yeah. Let's see. I'll have to navigate to the lecture. Well, do I? Nah. I'll try to explain it just on, just verbally. But if it's still not clear, let's come back to. I'll go to the lecture. So, polar bonds are generated between two atoms that have large difference in electronegativity. So, the higher the atom is on the electronegativity scale, the more it attracts the more it pulls the electrons towards itself. So in the polar bond, the example of a polar bond is the bond between the hydrogen and oxygen atoms in a water molecule. And oxygen is much more electronegative compared to hydrogen. So the oxygen will pull the electrons from the two atoms of hydrogen and have the partial negative charge, whereas hydrogens will have a partial positive charge because they don't have electrons, the negatively charged particles, close by anymore. They got pulled to the oxygen. And as a result, you have like one partially negatively charged side of the water and one partially positively charged side of the water. If the bond is nonpolar, in covalent nonpolar bond, it is formed between the atoms that are similar in their electronegativity properties. For instance, carbon and hydrogen are similar in their electronegativity properties. So the atom, let's say, of methane, the molecule of methane that contains one atom of carbon and four atoms of hydrogen will have non-polar covalent bond because electrons will not move neither towards the carbon nor towards the hydrogen. The electrons will stay put by their respective atoms. So there will be no partial charges within a molecule. Does that answer your question, Sarah?
Yeah, it does. So when I'm like writing my notes for that, could I like draw like two arrows going away from each other for polar and then two going towards each other for nonpolar?
Well, nothing goes towards anything in non-polar.
So the difference is just that it's like similar electronegativity?
Yeah.
Okay.
The difference is it's the similar electronegativity, but the similar electronegativity results in no pull of atoms anywhere.
Oh, okay, okay. Cool. Thank you.
Yep.
So I have another
Could you
question.
go over the effect of temperature and pH on enzyme activity?
Okay, Elena, so temperature and pH and enzyme activity. It's so remember the graph on x-axis there was temperature, on y-axis there was speed of reaction, and there was this graph that with temperature the speed of reaction increased and then at some point it dropped off. So this increase in reaction rate is mediated by the enzyme activity. It's the same. You can view it the same as enzyme activity. Enzymes speed up chemical reactions, okay? And the enzyme speed up chemical reaction at a higher efficiency. So enzymes are more active as the temperature increases. However, there is a certain critical temperature after which the protein denaturation occurs, where protein basically breaks down, loses its properties, and after which the enzyme loses its functionality and reaction just doesn't go anymore. And whenever the reaction doesn't go anymore as a result of too high of a temperature, you will see this sharp drop off in the reaction rate against at certain temperature. Okay, and pH also acidity also mediates enzyme activity. Like there's some optimal pH that can influence enzyme activity, but not all of the pH are. For instance, if the environment is too acidic, the reaction won't go, and if the environment is too basic, the reaction won't go. Like the optimal pH is often at least in our bodies, the optimal pH is like neutral in most parts of the body, except for the stomach. There, it's acidic. Did that answer your question, Elena?
Yes, thank you.
Yep.
On the topic of mitochondrial DNA and the genetics of that, is the only thing we really have to know just that it's maternally inherited? Because I looked over my notes and I feel like that's all I really found.
Yeah, that's right. So, like things to know about that mitochondria contain their own DNA, and that DNA is only maternally inherited because when fertilization occurs, so when the male gametes meets the female gametes, the DNA in the mitochondria in the male gametes, they get disintegrated, and only the mitochondria from the female gamete is preserved.
Okay, thank you. And also, kind of related to the mitochondria, do we need to know like the structure, like the cristae and the ribosomes that are in the mitochondria? It was shown in like that diagram on the lecture.
I don't. If it's not in the study guide, if the structure of the mitochondria is not in the study guide, I would not put much focus onto it.
Okay, all right, thank you.
So some of my other courses, they also use the lockdown browser, but we're still allowed to use our notes. They just don't want us to use the internet. Are we allowed to use our notes on the exam?
Nope. No notes. It's closed notes, closed everything. Guys, the study guide is pretty comprehensive. I mean, it shouldn't take you long, and I don't think the exam is too difficult. So yeah, I don't think you will even need your notes. But the point is, the bottom line is, it's closed book, closed notes, closed everything. So don't use your notes.
Okay, just making sure.
Professor, there's some questions about the time for the test and giving more time. Is that student accessibility services would have to contact?
Yes. So I cannot give you more time unless student accessibility services send me an email. Okay, so also another thing that I will follow up with an email. Please start the exam at 9:30 a.m. Okay, everybody should start the exam at the same time. If you start like 10 minutes later, I will.
Sorry, that was.
Yeah, it's okay. If you start 10 minutes later, I will suspect that you were cheating. Okay, so and I will look into it very closely, and look for evidence that you were cheating. So everybody start at 9:30 a.m. Do not start it late. All right, other questions?
Professor, I looked back at the study guide and there is a section that talks about the like structure of the mitochondria.
Okay.
So, I mean, what part of like the diagram, or would you just suggest that we review like the diagram that shows the intricacies of like the mitochondria?
Yeah, I guess this will be enough. So, outer membrane, inner membrane, cristae, which are the folds within the mitochondria. And there are proteins embedded in the membrane of the mitochondria that are part of the electron transport chain. And mitochondria have their own DNA.
Okay, thank you.
Professor, I have a quick question.
Go ahead.
I think Ansley sort of answered it, but I was wondering if do we start the test, like we click it at 9:30, and then it'll make us do the picture and also the recording around our room, or do we start that before and then we'll have the option to start the test exactly at 9:30?
Good question. So the test becomes available at 9:30 a.m. So go ahead and start it from the lockdown browser at 9:30 a.m.
Okay. The reason I ask is for the practice quiz that we did. Like we were in the lockdown browser already, and we had to click the quiz. And as soon as we clicked to start the quiz, it was making us do the picture, the ID, the recording of the room,
Yeah.
which will take us, I don't know, 5 minutes or so, I guess.
I don't think it takes 5 minutes. So when I did it, it took like maybe 2 minutes, and I'm overestimating it.
Okay. Cool. Thanks.
So we should just have our ID and everything just ready to go,
Yep.
so that it doesn't take as much time.
Yep.
Okay.
Yeah, treat it as an in-class exam, okay? So come ready. Don't cheat. Don't use notes. Don't use phones. Don't be on Discord. Don't be on what's this other thing? Discord and
Group me.
thank you. Yeah, don't be on GroupMe or other group chat servers.
Driver's license should work for the ID, right? As long as you can see their face on the ID.
That's right. So I think driver's license is less private than UCF ID. So, if you have both, give me UCF ID. But if you don't have UCF ID, then yeah, some picture ID will work. All right, it's 10:26. So are there any questions that were not answered yet?
I just have a general question about the exam. How many questions is it? If we're allowed to know.
I don't remember, frankly. I think it's about like 40-ish, maybe. Yeah, I don't remember, sorry.
Okay, thank you.
Anyone else? All right, guys, thanks for joining. Have a good weekend and good luck on the exam on Monday. All right.